there guys, in this video we will be taking a look at the digestive process in the stomach and highlight some of the key points that you need to know. Let's go through a quick review of the histological architecture of a hollow organ. Now, the innermost layer surrounding the lumen is called the mucosa. And the mucosa is composed of three parts: the epithelial layer, the lamina propria, and the muscularis mucosa. The next layer is called the submucosa, which is a connective tissue layer. And then there's a couple of layers of smooth muscle called the muscularis externa. And then around the outside of the organ we find either a serosa or an adventitia, depending on the location of the organ. The histology of the stomach is similar to the rest of the digestive tube in that we see the same four layers. However, there are some specializations in both the mucosa and the muscularis externa that are unique to the stomach. In this picture of the stomach, we can see that we've got three layers of the muscularis externa. We have the usual outer longitudinal layer, the inner circular layer, and then the innermost layer is an oblique layer that allows a greater mechanical breakdown of food. We can see the three layers of the muscularis externa very nicely in this next picture as well, along with all the other layers. I want to now focus on the mucosa for the rest of this video. Now, it's no accident that the layer directly surrounding the lumen of a hollow organ is called a mucosa, and the stomach readily demonstrates that point. The simple columnar cells of the mucosal epithelium of the stomach secrete copious quantities of alkaline mucus that forms a protective barrier. The mucosa also contains many gastric pits, which are invaginations of tubular glands that contain four different cells. And in the upper portion of each gland we find mucous neck cells that produce an acidic mucus. The second cell that we find is the parietal cell, which produces hydrochloric acid and intrinsic factor. Hydrochloric acid is important for the proper function of pepsin, the main digestive enzyme of the stomach, and intrinsic factor is needed for the absorption of vitamin B12 in the small intestine. The third cell that we see is the chief cell. The chief cell produces pepsinogen, which is the inactive form of pepsin, and hydrochloric acid is needed to activate pepsinogen to become pepsin. The fourth cell of the gastric gland is called the enteroendocrine cell, also known as the G cell. These cells produce a number of paracrines and hormones. We're going to focus on one of them called gastrin, that functions locally to control secretions and motility. Now, the mucosal barrier is exposed to very harsh conditions, and there are two main hazards to the mucosa. First is the acidic juice that is produced by the gastric glands. And the pH of the stomach ranges between 1.5 and 3.5. Direct contact with this acid could destroy the mucosal lining. The second hazard of the gastric juice is that it contains an enzyme that catalyzes the breakdown of proteins, and that's pepsin. So what prevents the stomach from digesting itself? As previously mentioned, the epithelium produces a thick mucus that is alkaline, and that's because it's rich in bicarbonate. This mucus functions as a physical barrier, as well as having a buffering effect to neutralize the acid that may be in close proximity to the mucosa. Another protective mechanism is that the epithelial cells have tight junctions between them. And this is protective in that it doesn't allow the stomach acid to penetrate into the deeper layers of the stomach lining. And the third protective mechanism is a rapidly dividing epithelium, which is constantly shedding and being replaced. Now, these protective mechanisms are very important because a breach in the mucosal barrier will cause inflammation of the stomach wall. And this is called gastritis. And persistent damage to the mucosa with damage to the underlying tissues can promote peptic ulcers. 90% of recurring ulcers is the result of a bacteria called Helicobacter pylori. This bacterium burrows itself into the stomach lining and causes tissue damage, but it's treated very easily with antibiotics. So I want to move on to the digestive process that occurs in the stomach and what is controlling it. So food in the mouth will begin the process of gastric secretion in the stomach. Well, actually, the process can begin even before the food enters the mouth. Just the sight or the smell of food can stimulate the vagus nerve, stimulating the gastric glands to begin to produce gastric secretions. This stimulation is referred to as a long nerve reflex because it is not initiated in the organ itself. Once food hits the stomach, then we have the short reflexes occurring. These reflexes are under both neural and hormonal control. In the stomach, the presence of partially digested proteins, caffeine, and a rising pH stimulates the release of gastrin from the enteroendocrine cells of the gastric glands. Gastrin, in turn, will stimulate the release of pepsinogen from the chief cells and hydrochloric acid from the parietal cells. We'll be taking a look at the feedback mechanisms at work with this process, but first I want to take a closer look at the parietal cell. All right, let's get ourselves oriented to what we're looking at here. So we've got the parietal cell, which is an epithelial cell within the gastric gland, and we have the stomach lumen and the apical membrane of that cell. And then we have the basolateral membrane of that cell, and then we've got interstitial space, and then capillaries. Let's look at the apical membrane of the cell, and we see that it contains hydrogen-potassium ATPase, and this is an antiporter, which means that the hydrogen and the potassium are going to travel in opposite directions. So as potassium is pumped into the cell, hydrogen will be pumped out. The hydrogen ions are produced by the breakdown of carbonic acid and yields a proton and a bicarbonate ion. The bicarbonate ion is going to exit the cell at the basolateral membrane, and we see another antiporter pumping out the bicarbonate ions in exchange for chloride. And the chloride then leaves the apical membrane and goes into the lumen, and this forms hydrochloric acid. So the bicarbonate ion is going to be picked up by the blood capillaries that are leaving the stomach, and the blood becomes more basic because of this, and it's referred to as the alkaline tide. Now don't worry, this will be counterbalanced by the blood exiting the pancreas, which tends to be more acidic, and that's called the acid tide. Now this production of hydrochloric acid is stimulated by three chemicals, and the three chemicals are acetylcholine, which is released by parasympathetic nerve endings, gastrin, that is released by the enteroendocrine cells, and histamine, which is released in response to gastrin. When one chemical binds to the parietal cell, hydrochloric acid secretion is minimal, but when all three bind, copious amounts of hydrochloric acid is produced. Antihistamines such as cimetidine block the histamine H2 receptors on the parietal cell, reducing the production of stomach acid, but not inhibiting it completely. On the other hand, direct inhibition of the pump itself will have a much greater effect at reducing stomach acid production, and these medications are referred to as proton pump inhibitors. So now it's time to put all this information together to see how this mechanism becomes self-regulatory. And we've got just a picture to show that. So we can see that the presence of food in the stomach elevates the pH, which stimulates the G cells. And the G cells will release gastrin, and gastrin stimulates both the parietal cells and the chief cells to release their secretions. So the chief cells will secrete pepsinogen, and parietal cells will secrete hydrochloric acid. And in the presence of hydrochloric acid, pepsinogen is going to be converted to pepsin, which is the active enzyme that catalyzes the breakdown of proteins. So as proteins begin to get broken down, they're going to cause a change in the acidic environment, making it more alkaline. And this again is going to start to stimulate the G cells to produce more gastrin, which perpetuates the cycle and keeps it going. So as stomach contents become more alkaline, enteroendocrine cells are stimulated to release gastrin, whereas if the stomach contents are more acidic, enteroendocrine cells are inhibited and do not produce gastrin. So what we see at work here is a wonderful negative feedback system that is maintaining the pH of the stomach at an optimal range between 1.5 and 3.5. So that's going to do it for this video. We'll pick it up with the small intestine next time.